All right, welcome everybody. We're missing one committee member, but I got a text that she is stuck in traffic somewhere, so who knows. So very pleased that everyone can be here. Rosalind and I are both day 4 without power. How about everybody else? Anybody else? Yep, a couple. Jose, Gary. So this is great that everybody's here. So I'm just really pleased to introduce Rosalind. Rosalind got her undergraduate degree at Cornell University in biology with a concentration in genetics. And then she worked for 2 years at Boyce Thompson. And I don't know if that was your first experience of working on plants and fungi together, but she worked on mycorrhizal associations with Medicago truncatula. And it must have been an amazingly productive time because 2 years of working, and she has 5 research publications and 1 chapter just from that 2-year stint, so that's quite something. She applied for and received a very prestigious NSF predoctoral fellowship, so we were really excited when she decided to come here, be a plant pathology student and work in my lab, and it was brave on her part because this was a new project that Gene Restaino and I had gotten some money from Dole Foods to work on this Amphigiensis banana interaction. It was a completely new project to my lab, and so then you're starting from scratch. So here you bring in one graduate student who's supposed to solve every problem, and she had to figure out just even how to transform this stupid fungus and all kinds of other techniques because she was starting from ground zero. And she has done quite an amazing job, as you'll see in her seminar today. Wrote down what I wanted to say. So far, 2 of her papers have been published, and a 3rd one we just heard yesterday has been accepted pending some revision. So she will talk to you about that. We also have lots of data on another project, which Elizabeth Thomas in the lab will be following up on when Rosalind goes on. In addition to her NSF, she also received funding through an NIH predoctoral fellowship. She did an internship at Monsanto. She's won a number of awards, travel awards from the Microbiology Society as well as from the Phytopathology Society. She's the author of numerous publications, so she's done a lot. Right now, she's just recently started a new position. She is a researcher with the Center for Integrated Pest Management on campus, and she is doing big data work to map how pathogens are spreading and emerging pathogens so that there can be early warning sent out on these pathogens that are spreading. So, Rosalind, we're really excited to hear about your work.
Thank you very much. So my dissertation has been about the pathogenicity mechanisms of Mycosphaerella fujiensis, causal agent of black sigatoka disease of banana. So bananas are one of the most important crops in developing countries, and they're important both as an export crop and for subsistence farmers. And most bananas are actually grown for subsistence farmers. Edible bananas, the seedless varieties, are typically hybrids or polyploids of 2 wild banana species, and these have mutations that make them seedless and parthenocarpic, which means that the fruit develops without pollination and fertilization and without the seeds developing. Here you can see what seeds in a wild banana would look like and why you would really want to have a seedless banana variety to eat. And so because of this, bananas have to be propagated vegetatively, and this can be done in a couple of ways. You can propagate them the traditional way by letting the banana plants sucker, so here's a little banana sucker, and then you can dig these up and you can plant them in new fields wherever you want them. The downside of this method is that whatever diseases might be on these plants, you're then spreading that from field to field. And so for that reason, commercial banana plantations have gone to tissue culture. So here's what banana tissue culture looks like. And here you're growing bananas in a sterile way in culture. And so you're minimizing the spread of disease. But still, since you're having this clonal organism, this banana, disease is a big problem. And black sigatoka is considered the most costly disease of bananas. So this is caused by a fungus, Mycosphorella fijiensis. It was first described in Fiji in 1963, and since then it's spread to basically all of the banana growing regions around the world. You can see what the symptoms of this disease look like. You have these necrotic areas on the leaf, and as you can imagine, these plants are not able to undergo photosynthesis as well as they should be. And so that reduces the yield by itself, but also the disease causes the fruit to ripen prematurely. This is a huge problem for export companies because they think they have some nice bananas that they can ship out, but by the time they reach their destination, they can be overripe. So to combat the disease, fungicides are being used, and these are being sprayed extensively, in some cases every 5 days. And this is comprising about 1/3 of the total production cost of banana, just dealing with one disease. So clearly, we need new ways of dealing with this pathogen. This is the life cycle of the fungus, Mycosphorella fijiensis. So you have asexual spores, conidia, or sexual spores, ascospores, which can go and land on the banana leaves. These will germinate in wet weather, they infect through the stomata, and they cause necrosis of the leaf. And then the fungus will go through its asexual cycle to produce more conidia, and through its sexual cycle to produce more ascospores. And both of these can spread and infect new plants. And ascospores in particular can spread very long distances, so kilometers, and on the wind and infect new plants. There's not a lot known about the pathogenicity mechanisms of M. fijiensis, but I'm going to briefly tell you a little bit about what was known prior to my work. So pathogens in general, they use effectors. These are small proteins that are secreted, and they modulate the host defenses and they shut that down. So pathogens typically have a lot of these effectors, and in Cladosporium fulvum in particular, there are these 3 effectors known, AVR4, ACP6, and ACP2, and they're homologs of those in M. fijiensis. So those may be involved in disease, but we don't know exactly how important they are, and furthermore, that's likely to represent only the smallest tip of the iceberg for the effectors. In Fusarium species, it's known that nanoproteins in the fungal cell wall are important, these GPI proteins, and there are homologs of these in M. fijiensis. So again, perhaps they're important in disease, we don't know exactly. In a closely related species, Mycosphorella graminicola, there's an ABC transporter that's important for colonization in the substomatal cavity, so this is right after the fungus penetrates into the host. And there's a homolog of this in M. fijiensis. Again, same story, we don't know exactly the role in M. fijiensis. And finally, because of the necrotic leaf lesions that are observed in black sigatoka, it's long been thought that toxins are involved in the disease process to kill that host tissue. And there have been several studies try to look at what types of toxins would be involved in that. And primarily, these have identified melanin shunt metabolites. And furthermore, in other fungi, melanin itself is known to be important for pathogenicity. So it was thought for a while that this was one critical pathway for pathogenicity in M. fijiensis. However, this pathway was recently disrupted and shown to not affect pathogenicity. So clearly, we don't know a whole lot. So that's where my research comes in. And because of this focus on toxins as a possible pathogenicity mechanism, I have been focusing on secondary metabolites, and in particular, polyketides. So a lot of these are important pathogenicity factors in different plant pathogenic fungi, and here I have several examples of important polyketides from different fungi. You may recognize the names of some of these, but in particular, I'm going to point out these 2. So Cercospora and Ramularia species are very closely related to Mycosphorella fijiensis, and these produce polyketide toxins called cercosporin and rubellin. These both work in a very similar way. So, for example, here's cercosporin, and rubellin is somewhat similar to this, but in the light, it becomes a photosensitizer. So it's producing reactive oxygen species, which then go and kill the host tissue, leading to the symptoms that you observe. And in Cercospora species, light is known to be essential for pathogenicity. Similar to that, with Black Sigatoka, it's known that if you grow the plants in partial shade, it takes longer for the symptoms to develop. So we think there could be a similar toxin in Amphigiensis, and that's why I'm focusing on polyketides. So that brings me to the data from my first paper, which was published this summer, called Bioinformatics Prediction of Polyketide Synthase Gene Clusters from Mycosphorella amphigiensis. So the Amphigiensis genome sequence has been publicly available for several years now, and I was able to use bioinformatics predictions to look at what polyketide synthase genes are present in the genome. So using bioinformatics, I predicted that there were seven polyketide synthases and 1 hybrid enzyme. So this has features of polyketide synthases and non-ribosomal peptide synthases, which are another type of secondary metabolite. And all of these are large multi-domain enzymes. So each domain has a different job to do in the production of the polyketide. And you can predict what domains are present, and based on that, you can predict what type of product is likely to be produced. So these are the domains that were present in each of the putative enzymes that I was identifying. And specifically, there are domains that are critical for the function of all polyketide synthases, and there are other optional domains. If you look at the optional domains, you can predict the degree of reduction of the polyketide product. So over on the right, these are polyketides produced by other fungi that are produced by non-reducing enzymes. So you'll see a lot of aromatic rings and a lot of double bonds in these structures. On the left, these are polyketides produced by other fungi where the polyketide synthase is a highly reducing enzyme. So you'll see more linear structures with a lot more single bonds in comparison. So when I looked at the optional domains predicted from each polyketide synthase in Amphigiensis, there were 3 enzymes predicted to be non-reducing, so PKS 7.1, 8.1, and 10.1. So their product should be something like this. There were 2 that were highly reducing, PKS 2.1 and 8.2, so we would expect their products to be something like this. And there were 3 that were partially reducing, so those should be somewhere in between those 2 extremes. So the next thing I wanted to do was look at the polyketide synthase sequences and predict, well, are any of them homologous to anything that's been characterized from another fungus? So to answer that question, I generated this phylogenetic tree, and I'm just going to point out a few different clades of interest. So here, this clade has a lot of melanin-producing polyketide synthases from other fungi, and the Amphigiensis PKS 10.1 is in that clade with them. So this PKS 10.1 should be the melanin-producing polyketide synthase in Amphigiensis. Down here is a clade with the fumonisin-producing PKS from Fusarium, and this clade also contains the PKS 8.2 sequence from Amphigiensis. So we would predict that those 2 products should be somewhat similar to each other. Here, PKS 2.1 from Amphigiensis is in a clade with PKSs producing alternopyrone and T-toxin. And here, PKS 10.2 is in a clade with a PKS producing solanopyrone. But so far, I've just been looking at the polyketide synthase sequences, and there are other enzymes that are going to modify that polyketide to produce the final product. Now, in fungal genomes, all of these genes tend to be clustered together in the genome, so I can look and see what genes are adjacent to the polyketide synthase gene, and also make further predictions about the type of product produced by each thing. So when I did that analysis, here's what I found. So up here, these are the different polyketide synthases that were predicted from the Amphigiensis genome. And over here, these are the type of genes that would be predicted from these types of clusters. So you can see that each cluster is containing several different types of genes that you would expect. So then we can look and we can compare different gene clusters to learn more. And here I'm just showing you one example of where I did that. So in the phylogenetic tree, I had showed that the PKS82 sequence is most similar to a sequence of a PKS producing fumonisin. So now I'm comparing the whole gene cluster. And up here at the top, this is the fumonisin producing gene cluster from Fusarium. Down here, this is the PKS82 gene cluster from Amphigiensis. Genes that have similar predicted function I'm showing in the same color. Genes with different predicted function I'm showing in light blue. The first thing that will jump out at you is that the fumonisin biosynthetic cluster is much bigger. So it has a lot of genes that Amphigiensis doesn't have in its gene cluster. However, fumonisin is known to act as a sphingosine analog. So it inhibits sphingolipid biosynthesis in the plant. And knowing that, if we look at what genes are in the PKS82 gene cluster, they are genes with homology to sphingolipid hydroxylase and ketosphingosine reductase. Also, these two clusters share an alpha-oxoamine synthase, and in fumonisin biosynthesis, it's known to add this amine group that makes it more similar to sphingosine and therefore able to perturb sphingolipid biosynthesis. So although the clusters are different, we can still conclude from this that perhaps they're producing a product with a similar function. So that analysis was depending on me knowing something about fumonisin and its role in biology. But I can also do this type of analysis where I'm looking at Amphigiensis gene clusters and comparing them to unknown gene clusters in other species and looking at conservation of that gene cluster. So there are actually 3 pathogens that cause similar disease on banana, although Amphigiensis is considered the most important one. And if I look at gene clusters in all of them, I'm showing you one example here. This is the PKS10-2 gene cluster. Here's the gene cluster in Amphigiensis. And here's the cluster and its close relatives, Mycosphaerella musicola and Mycosphaerella eumusae. You can see this gene cluster is highly conserved, and I was unable to identify this gene cluster from other close relatives of Amphigiensis that are not pathogenic on banana, but are pathogenic on other things. So this was really interesting. And we don't know definitively that this would be a pathogenicity factor for disease on banana, but it's an interesting hypothesis. Another way I looked at this was looking at expression of the genes. Because if a polyketide synthase gene is important for pathogenicity, it should be expressed at least some of the time when the fungus is infecting its host. You wouldn't necessarily expect it in culture, though. So I did RT-PCR assays under both conditions. And here, this is showing the expression in the leaf. You can see there are several polyketide synthase genes that are expressed under these conditions. There are also some polyketide synthase genes expressed in the media. What's really interesting from this, though, is this PKS10-2. This is expressed in most of my infected leaf samples, and it's not expressed in the media. I looked at this another way. So this is using RNA-seq. This is a different isolate of the fungus, and I'm trying to validate my earlier findings and also learn more, because now I can look at all the genes in the cluster at once very easily. So this is showing one example. This is the PKS82 gene cluster. The genes predicted from the cluster are shown in purple right here, and the black bars are showing gene expression in the leaf versus the media. So these bars above the line are representing genes with higher expression in the infected leaf tissue compared to the media. You can see that most of the genes from this cluster do have higher expression in the infected leaf tissue. So this was a really interesting finding, and it enabled us to learn more about the possible boundaries of this gene cluster. Just going to show you a couple other examples here. Here's another gene cluster, the PKS10-2 gene cluster. This also had much higher expression in the infected leaves compared to the media, but not all of the gene clusters had higher expression in infected leaves. Here's the melanin gene cluster that had lower expression in the infected leaves, so they had higher expression in the media. So from this first paper, here are my conclusions. I identified 7 putative polyketide synthase and 1 hybrid gene. I showed that PKS10-1 gene cluster is likely involved in melanin production, and in fact this has been verified experimentally. And I showed that PKS21, 82, and 10-2 are similar to enzymes producing alternopyrone, fumonisine, and solanopyrone. I did a comparison of gene clusters and showed that the PKS82 product is likely to have a similar biological function as fumonisine, although perhaps those products are chemically different from each other. I didn't show this, but it was a little less conclusive for the other gene clusters. I showed that some, but not all, gene clusters conserved in other Sigatoka pathogens. And I showed that the PKS10-2 cluster is conserved in the Sigatoka pathogens, but not a lot of other close relatives. And the RNA-seq and RT-PCR analyses showed some, but not all, of the PKS genes having higher expression in infected leaves compared to media. So that brings me to the second part of my talk. So this is going to be a paper. We submitted this, and we just got response back that it will be accepted pending revision. This was done with my collaborators, so Dr. Shi, Morgan Carter, Dongmi Ma, and of course my advisor, Dr. Daub. This will be entitled "A Polyketide Synthase Gene Cluster Involved in the Sexual Reproductive Cycle of the Banana Pathogen Mycosphaerella fijiensis." So there was a paper a while back that showed that in Neurospora and Sordaria species, there's a polyketide synthase that's important for sexual reproduction. In particular, female fertility and development of paresthesia in those fungi. So I wanted to see if any of the polyketide synthases are homologous to those. So I generated a new phylogenetic tree, and I'm going to point out this clade right here. So PKS84 and the hybrid 83 enzyme were coming in a clade with the PKS enzymes from Sordaria and Neurospora. And also in this clade were enzymes that produce compactin and lovastatin. And while compactin and lovastatin have been studied extensively for their role in modern medicine, these are less well characterized for their normal role in fungal biology. But what is known is that these are produced during the sexual cycle of those fungi. So my hypothesis here was that perhaps this whole clade could be involved in sexual reproduction in these fungi. And I wanted to characterize at least one of these enzymes from Emphigiensis. So I mentioned before that polyketide synthases are large multi-domain enzymes. And so one analysis that I did here was look at the predicted domains from each of the polyketide synthases. So the ones from Emphigiensis, the ones from Sordaria and Neurospora, and the ones producing compactin and lovastatin. So these are the ones from Neurospora and Sordaria, and you'll see that they're more similar to the PKS84 enzyme than they are to this hybrid enzyme. So that right there was saying maybe I want to look more at PKS84. I also looked at these expression assays. So I showed you this one before. So these are my RT-PCR assays of infected leaves versus in culture. And this hybrid PKS and RPS gene, it seemed to have pretty consistent expression both in the infected leaf and in the media. However, the PKS84 gene seemed to have maybe a little bit higher expression in the infected leaves compared to the media. And when I looked at my RNA-seq data, both of these clusters had higher expression in infected leaves compared to media, but the difference was greater for PKS84. So here's this log2 fold change, 4.4 compared to 2.3. So I wanted to characterize PKS84 because of these analyses. So one way that I did that was by creating a PKS84 promoter GFP fusion and transforming that into the Emphigiensis. And so I did that, and I observed it under conditions promoting mating as well as other conditions. So these These are small circular areas of GFP fluorescence that I was observing primarily under mating conditions to promote mating. And then I looked at a higher magnification on the confocal microscope, and this is what these structures look like at that higher magnification. So then if we look at the life cycle of a closely related Mycosphaerella species, you have the vegetative mycelium, and when it's going into its sexual cycle, it will produce the female structures, so these are the proto-pseudothecia, and it will produce the male structures called the spermagonia. The spermagonia are filled with very, very tiny cells called spermatia that exit through the spermagonium and fuse with the female structure, the trichogyne. And the female structure will continue to develop and form the pseudothecium and go through meiosis and produce the ascospores. So what I was seeing there was very consistent with the expected structure of these pre-pseudothecia. So then I wanted to look at different stages of development and see, okay, is this promoter active throughout the development of pseudothecia? So here's a picture of an early stage of development. So on the left, this is a 3D projection of the GFP fluorescence. And on the right, this is a single optical slice, and it also shows the transmitted light. So it's active at early stages, and here's an example of what it looks like at a later stage. On the right, if we look at just the transmitted light, you can see these are very highly melanized, so it's much more developed at this point in time. And this promoter is still very active, so you can see all this GFP. So next, I generated a mutant of this polyketide synthase gene, and I showed that the disruptant was still pathogenic. So here is a plant inoculated with the disruptant. You can see these small necrotic areas characteristic of Flexigatoka. And on the right, this is not disruptive for this gene. So it's still pathogenic. And then, unfortunately, I was only able to get one mutant, and I had to mate that with a wild-type isolate of the opposite mating type. And under these conditions, it still produced the sexual structures. You can see these black dots for both the wild type and the disruptant. However, if you remember back, I was saying that in Neurospora and Sordaria, this gene is important for female fertility. So we don't know definitively if this gene is important for female fertility in this fungus, because it could be that the wild type fungus of the opposite mating type is acting as the female parent. So my collaborators also helped in doing some metabolite profiling of this mutant, and we were able to show that the mutant here has a very different profile from the wild type. So each of these are showing different metabolites, and the patterns are more similar for mutant versus wild type. And in particular, we showed that there were perturbations of the fatty acid esters, and fatty acids and polyketides have the same precursors. So this was not unexpected. So my conclusions from this paper were that the PKS8-4 and hybrid 8-3, they're in a clade with PKS enzymes involved in sexual reproduction. PKS8-4 has more similar domains than the hybrid, compared to the Neurospora and Sordaria sequences. PKS8-4 and hybrid 8-3 both have higher expression in infected leaf tissue, but the difference is greater for PKS8-4. The PKS8-4 promoter is active in developing pseudothecia at different stages of development. The mutant of this gene is still pathogenic, and it still produces pseudothecia if it's paired with an isolate of the opposite mating type. We don't definitively know if there's an effect on female fertility, however. And the mutant has changes in the metabolite profile, including changes in fatty acid esters, which may be consistent with a knockout in the polyketide synthase. Okay, so for this part, this is another paper where I'm focusing in on a particular polyketide synthase. And here, the authors on this paper will be me, Elizabeth Thomas in our lab, Dr. Shi again, and of course, Dr. Daub. This is still in preparation, but the title will be A Novel Polyketide Synthase Gene Cluster in the Plant Pathogenic Fungus, Mycosphaerella emphigiensis. A lot of polyketides, including cercosporin and rubellin, which are produced by the closely related fungi, a lot of polyketides are fluorescent molecules. And so one way I was looking at polyketides produced by emphigiensis was to screen through colonies of emphigiensis and look for fluorescence. And in fact, I did observe red fluorescence occasionally in emphigiensis cultures. So here you can see what this looks like. And I wanted to characterize this red fluorescence. And so I used confocal microscopy to generate fluorescence emission spectra. Over on the left, this is emphigiensis. This is from cultures fluorescing red, and up at the top, these cultures were not fluorescing red. They appear to have a little yellowish autofluorescence, but under high magnification, you can see there are actually areas of green fluorescence and what looks like maybe some very, very faint red fluorescence in some areas. Now, for Cercospora producing cercosporin, it actually looks very similar. So you see these hyphae, they fluoresce green, and then there are areas of red fluorescence. And this is consistent with the cercosporin fluorescence because depending on whether it's oxidized or reduced, it can fluoresce red or green. So I wanted to generate fluorescence emission spectra for both the green and red fluorescence from both fungi. And when I did this, I found very similar fluorescence emission peaks. I also looked at the solubility of the red fluorescent compound from emphigiensis, and I showed that it has similar solubility as cercosporin does. And then I looked at the absorption peak. So this is showing here's the emphigiensis absorption peak from these extracts, and here's the peak from Cercospora nicotiana. So this is from cercosporin. You can see these are different absorption peaks. However, in the emphigiensis absorption peak is similar to absorption peaks of compounds that are similar to cercosporin. So it could be a similar compound. We don't know definitively. Then from those extracts, I identified 14 new compounds with my collaborators from the red fluorescing emphigiensis extracts. And 3 of the compounds were similar to compounds reported from polyketide pathways. So here are the structures of those compounds. Then I looked at, well, is there any correlation between the red fluorescence and expression of either polyketide synthase genes or genes in the putative gene clusters? And in fact, there appears to be some correlation for this transporter that's in the PKS81 gene cluster. So it looks like it's expressed when there's red fluorescence and it's not expressed when there's not red fluorescence. So this focused me in on this PKS81 gene cluster. Wanted to do further analyses on that. So one analysis that I did was to look at conservation in close relatives of emphigiensis. And in fact, I showed that this cluster is conserved. So it's a very large gene cluster. You can see here it is in emphigiensis. Here's the M. musicola and M. eumusae. It's also conserved in other relatives that are not pathogenic on banana. So they're pathogenic on other things. So here's an example of that. And then recently there was a paper that came out that said that there is a homologue of, so from emphigiensis, and a homologue of this PKS that produces monodictaphenone. So I wanted to investigate that and see, okay, which PKS is it? And in fact, it did come out in this clade with PKS81 from emphigiensis and its homologues from these other Sigatoka pathogens. So then if we compare the structures of monodictaphenone and some of the things that I identified earlier, So here's monodictyphenone produced by Aspergillus fungi, and this cluster in Aspergillus can also produce emodin and endochrosin, depending on what genes exactly are there, what strain, and what's expressed. And from Amphigiensis, we had identified this gene called pulverochrominol. So this is a fairly similar structure. So we don't know definitively, but perhaps this is what's being produced by this cluster. So then I compared the PKS81 gene cluster for Amphigiensis and Aspergillus angelans. So this is a cluster that produces monodictyphenone and those other compounds. The genes with similar predicted function, these are shown in the same color, and genes that are different are shown in black. So you can see that these do share several genes, but there are also major differences in these gene clusters. So we would not predict that they'd produce exactly the same thing. We'd predict maybe some similar compound, though. So then, since I had showed that there was correlation with the transporter from the cluster and the red fluorescence, I wanted to overexpress that transporter, and also there's a transcription factor in this cluster that has some similarity to AFLR from aflatoxin production, and so I overexpressed that as well. And this is showing gene expression, so these are qPCR results. Over here, this is the transcription factor from the cluster. This is what I overexpressed. And in blue, this is showing the vector-only control compared to wild type, which is set to 1. And then the transcription factor overexpressor, you'll see this comes out at about 10-fold. So we were able to overexpress that about 10-fold, which is what we wanted to see. And then for the other genes in the cluster, we did get overexpression of some of them. It was more modest. So here's PKS81. You can see there's a difference between the overexpressor versus the wild type and the vector-only control. However, we didn't get overexpression for all the genes in the cluster. So for example, this AFLJ-like gene, that's about the same expression. And then we have aldoketoreductase reductase over here, about the same expression. So that was a little disappointing that we are not able to get overexpression of all the genes in the cluster, and we are also not able to see red in these overexpression clusters, but it may be due to the fact that we couldn't get overexpression of everything. So it's possible we were accumulating some intermediate compound. So then we did inoculation experiments looking at the transcription factor overexpressor. So here's the overexpressor inoculated onto banana. This is the wild type and the vector-only control. So this is what it looked like. We did this 3 times. This is what it looked like in 2 of them. So you'll see that the overexpressor caused a lot more disease here compared to the wild type. Unfortunately, in the 3rd experiment, the disease was just so variable each time, and that time we got similar amounts of disease. So we don't know definitively what's going on. Further research is ongoing. From this paper, we can conclude that the red fluorescence is occasionally observed in Amphigiensis colonies. This has similar fluorescence emission spectra to cercosporin. It has a different absorption peak from cercosporin, but it's in the range of similar compounds called quinones. We identified 14 new compounds from Amphigiensis red fluorescent extracts, and 3 were similar to metabolites produced by other polyketide pathways. The red fluorescence correlates with expression of a transporter gene from the PKS81 gene cluster. PKS81 is in a clade with the monodictyphenone producing PKS. And the PKS81 and monodictyphenone clusters share some genes, but there are major differences also. And the overexpression of the transcription factor upregulates many, but not all the genes in the cluster. And unfortunately, I did not see any increased red fluorescence in those colonies. And there was variable results with inoculation. Okay, and then for the last part of my talk, this was also a paper that is published. This is called transcriptome sequencing of Mycospirilla apigensis during association with Musa acuminata reveals candidate pathogenicity genes. So before I get into this, I want to tell you a little bit about the transcriptome analyses that have been done prior to my work. And these analyses have almost exclusively focused on the banana side of the interaction. So there was a microarray of a susceptible versus resistant banana variety, and the resistant variety showed higher expression of phenylalanine ammonium lyase, peroxidase, disease resistance response 1, PR4, and PR10. There was a suppression subtractive cDNA library of inoculated or uninoculated resistant banana varieties at different time points. The upregulated genes after exposure with amphigiensis encode putative transcription factors, the NBS-LRR protein RGA1 and cytochrome P450 pathways, and changes in gene expression. There were changes in gene expression for the glycolysis gluconeogenesis pathway and genes for the scavenging of reactive oxygen species. There was also a suppression subtractive cDNA library of a susceptible banana, and the upregulated genes were involved in phenylpropanoid synthesis, detoxification, synthesis of ethylene, activation of jasmonic acid, and flavonoid and isoflavone-related genes. The only thing that was really done for amphigiensis is identifying expressed sequence tags of amphigiensis that were grown in a few different media. So even this experiment, it was not in association with its host. So that's why I wanted to do some transcriptome analysis from the pathogen side of things. So I've already mentioned that I did the RNA-seq analysis, but let me just explain it again. So I was comparing gene expression in infected leaves, like this, versus the culture medium. And down here, this is showing a principal components analysis plot. And this is just showing that the gene expression was very different for samples that were in the media, so these blue dots here, versus in infected leaf tissue. So this was exactly what I wanted to see, and this was helping to validate these assays. This is showing a graph of secondary metabolism related genes. So down here, each one of these is a type of gene that is commonly associated with secondary metabolism. And these green bars are showing the number of genes that had higher expression in infected leaf tissue. And the blue bars are showing the number of genes with higher expression in the media. So you can see for most of these, there are larger bars, larger green bars for these secondary metabolism genes. So there were a lot more cytochrome P450s that were upregulated, that had higher expression in infected leaf tissue, short-chain dehydrogenases, etc. So this was consistent with all the analysis so far, where we were looking at secondary metabolism and polyketides. But this is more broadly. This includes other types of secondary metabolism. Here, this is a graph of genes that have been associated with pathogenicity in other fungi. So, for example, cutinases, genes encoding proteins with the CFIM domain, genes encoding hydrophobic surface binding protein, and salicylate hydroxylase here. Again, you're seeing kind of the same thing. So there are a lot more genes that have higher expression in the infected leaf tissue compared to the media. This is showing miscellaneous types of genes down here, but what I'm going to point out to you is this bar here. This is showing genes encoding a domain of unknown function, 3328. So as the name suggests, we have no idea what this does. No one's really studied it before. But there were 11 of these genes that had higher expression in infected leaf tissue, and none of these genes had higher expression in the media. So perhaps this is involved in disease. We don't know definitively. Then I wanted to look at putative effector genes. So let me just refresh you. When the fungus is infecting the host, it's growing between the cells, and it needs to secrete effectors to modulate the host defenses and shut that down. So here's the fungus, it's secreting effectors. Some of them will stay in the apoplast, some of them will be taken up into the host cell. These effectors have certain common characteristics. They tend to be small secreted proteins that are cysteine rich. So the cysteine rich aids in stability when they're in the apoplast. And so I looked at genes encoding proteins with these characteristics, and I identified 30 genes with higher expression in infected leaf tissue that had those characteristics. Half of those had homologs only within the Mycosporulaceae, which is consistent with observations that most effectors have a restricted phylogenetic distribution. And most had no conserved domains and no homology to other characterized sequences. Finally, I wanted to look at a hypothesis that had been set forth by some other authors when the Amphigiensis genome was first published. So Mycosporella graminicola, this is a close relative of Amphigiensis, it's known to have 8 dispensable chromosomes. And dispensable chromosomes are chromosomes that are not required for survival of the fungus, but that can be important depending on the conditions that the fungus is growing at. And these dispensable chromosomes in M. graminicola had certain characteristics. They're small, they have a low GC content, they have different codon usage, there's a high proportion of repetitive DNA, there's a low gene density, and there's a high proportion of hypothetical genes. And it was shown that Amphigiensis has 14 putatively dispensable scaffolds with the same characteristics. So scaffolds are basically like chromosomes, but they're predicted bioinformatically, so we don't know for sure if they could actually be part of the same chromosome. So I'm just showing you a graph of one of these characteristics in Amphigiensis. So this is showing gene density. And you can see it's a really different value for the putative core scaffolds versus the dispensable ones. So it's quite dramatic, these differences. So I wanted to use my RNA-seq data to see if I could provide any support for this hypothesis. And the way that I did that was I created this graph. So over on the left, this is showing the putative core scaffolds versus on the right, the putative dispensable scaffolds. And these bars are showing the percent of genes on each scaffold that had higher expression in the infected leaf tissue, that's the green, or higher expression in the media, which is the blue. You'll see there's a very different pattern of gene expression on the putative core versus dispensable scaffolds. So on the putative core scaffolds, you're seeing a small proportion of the genes have higher expression in infected leaf tissue, and another small proportion have higher expression in the media. If we look at the putative dispensable scaffolds, it's very different. So you see either genes with higher expression in the infected leaf tissue, or genes that are expressed in that have higher expression in the media, or no differentially regulated genes, but never both on the same one. And in particular, I want to point out these two scaffolds. These had a very large proportion of genes that had higher expression in infected leaf tissue. So this one had about 1/3 of its genes. They had higher expression in infected leaf tissue. And for this one, it was over half of the genes on the scaffold had higher expression in infected leaf tissue. So perhaps this one could be involved in pathogenicity. We don't know definitively, though. And then I noticed that, so there were several that where there were not any differentially expressed genes. And when I looked more closely at this data, I saw that there were no transcripts at all that mapped to 2 of the scaffolds. So for 2 scaffolds, no genes were expressed. And for this RNA-seq data set, I used a different isolate of the fungus than the one that was sequenced. So then I wanted to know, well, are these even present in my isolate? And so I did some PCR assays, and it turns out that they're either absent or large stretches of them are absent, which is consistent with the hypothesis that these would be dispensable. So from this paper, I showed that genes involved in secondary metabolism have higher expression in infected leaf tissue. There were other putative pathogenicity genes that had higher expression in infected leaf tissue, and these encode hydrophobic surface binding proteins, CFEM domain containing proteins, and salicylate hydroxylase containing like proteins. I showed that 11 domain of unknown function 3328 genes have higher expression in infected leaf tissue, and none of these had higher expression in the media. I identified putative effector genes with higher expression in infected leaf tissue, and I was able to provide support for the hypothesis that Amphigiensis has dispensable chromosomes. So there were 2 of the putative dispensable scaffolds that have a large proportion of their genes with higher expression in the infected leaf tissue. And another 2 putative dispensable scaffolds may be absent or largely absent in the isolate that I used for RNA-seq. So the overall conclusions from my project, I was able to identify 7 putative polyketide synthase and 1 hybrid enzyme. There's the PKS10-1 gene cluster seems to be responsible for melanin production. PKS21, 8-2, and 10-2 are similar to enzymes producing alternopyrone, fumonisin, and solanopyrone. And there were several PKS genes that had higher expression in infected leaves. For PKS84, I showed that it's similar to enzymes important for female fertility in other fungi. It's expressed in developing pseudothecia. The mutant of this gene is not affected in pathogenicity, and it is altered in nonpolar metabolites. For PKS81, I showed that the cluster was similar to a monodictaphenone producing cluster. The transporter from this gene cluster has higher expression in red fluorescent colonies, and we were able to identify 14 compounds from extracts of the red fluorescent compound, including 3 that are similar to products of polyketide pathways. And actually, none of these 14 compounds have been previously reported from Amphigiensis, so it's exciting regardless. Overexpression of the transcription factor results in higher expression of some, but not all, the genes in the cluster. And my RNA-seq analysis identified genes with higher expression in infected leaves. These included secondary metabolite genes, genes encoding CFEM proteins, salicylate hydroxylase-like ones, hydrophobic surface binding proteins, effector-like proteins, and this domain of unknown function 3328. And the experiment also provided support for the hypothesis of dispensable chromosomes in Amphigiensis. Finally, I'd like to acknowledge my committee members, my lab members, other collaborators, my friends and family, my husband Jesse, my parents, and my fellow grad students. And finally, my funding sources. So I was funded by Dole Food Company, and as my advisor mentioned, the National Science Foundation and National Institutes of Health for my fellowships. Thank you so much.
Questions for Rosalind?
A question on
Yeah.
the dispensable chromosomes on the Nick one. Was that proven biologically that they are dispensable?
Yeah, it was. They were able to show that there are conditions where they lose them in culture, especially. I think when it goes through its sexual cycle in particular, it can lose them. It still grows just fine.
Following up on that, can they be reacquired by the fungus? And
That's a good question. So we don't know for sure. However, in Fusarium, it's been reported that they can transfer their dispensable chromosomes from one isolate to another and confer pathogenicity that way. So it's very well possible. We don't know.
I have a question. Your first set of experiments where you saw higher expression on banana leaves,
Mhm.
did you ever generate any knockout of these? And if so, what were the
It's so
results?
hard
Yeah,
to get knockouts.
I figured that you. I'm just.
I was
curious.
trying to get knockouts of a lot of the polyketide synthases, and I screened over 1,000 colonies of different different things I was trying to knock out, and I was only able to get one knockout mutant, so that was this PKS84 mutant that I was showing you. So I would have loved to do more, but yeah.
I was just wondering where that.
Yeah.
So great seminar. I really enjoyed the way you took your data and used it in multiple different ways, you know, and asked different questions. So I have a technical question and a big picture question. What would you like
Okay.
first?
Whatever you want.
Okay. Well, you know, the big picture question, and this doesn't always have to be, right? But where do you see this going in terms of disease control?
Well, the role of my research, I think, has been mostly to generate a lot of hypotheses, because so little research has been done from this fungus side of things. But if there were a gene that were shown to be really critical for pathogenicity or for disease spread, as may be possible for this PKS84 that might be involved in sexual reproduction, perhaps host-induced gene silencing could be used, so generating a banana that could silence these genes. So that's one way. Or if there were, if you could inhibit some of these pathways, which it's been shown that at least for melanin production, you can do that. So there's a compound called tricyclazole, which can control some other fungi, and that inhibits melanin biosynthesis. So perhaps it could be done chemically. You know, further research is necessary, as always.
That's right. So then the technical one on that PKS, where you did an overexpression, that was 8-1, I think you said?
Yeah. Mm-hmm.
Did you do those functional pathogenicity assays in the light and the dark?
The pathogenicity assays that I was showing you, those were done in the phytotron.
Yeah.
And so theoretically it should have similar light. However, the first two experiments that we did looking at pathogenicity, those are done in the summer, and we saw a bigger difference. And the last one, where we didn't see much of a difference, that was done in the fall. So further research is needed there to figure out, you know, is it the weather? I mean, I'm sure there was brighter light in the summer, so could have been that. We don't know. Yeah.
I just follow him. I want to praise you again. I mean, that's a wonderful seminar.
Thank you.
It's beautiful. At least easy for me to follow. You give me good, really big pictures about PKS. It used to be I think I misunderstood PKS. So it's really about I want to discuss. You know, we have many discussion about mutant PKS84.
Right.
I think it's actually. You say that this mutant does not lose the pathogenicity to the banana leaf.
It's still pathogenic.
Yes,
but,
they are pathogenic.
right.
Yes, that means another PKS possibly involved. Look at
Right.
the domains there. I can see there are a lot of compounds from other PKS.
Right.
So what I want to here is my comments that for you to think about the future. Since many these kind of polyketide compounds, plants do not make them.
Mhm.
So plants most of the time, most of this polyketide compound, they can cause some cytotoxicity to plant cells.
Right.
So plant cells most of them have a system, as you mentioned, a transporter. They can transport their own compounds from the cytosol or maybe from the plastid and the mitochondria to vacuum.
Mm-hmm.
The bigger vacuum is a storage site for
Right.
plant
Right.
to detoxify those compounds made by plants.
Mm-hmm.
So plants make a lot of compounds poison to animals, many fungi.
Right.
So when you have a pathogen, when the fungi infect them, make a polyketide,
Mhm.
many polyketide, the plant may not have a transporter to deal with them.
Exactly.
So then just cause the cytosol poison and the cause.
Right.
So I was wondering if we're going to transform, introduce your fungi as transporter to the plant, how the plant will deal with
That would
or be
worth
able to
trying,
deal with
yeah.
pathogens.
Interesting.
Yeah, that's what I'm thinking. Yes,
Right.
because look at many most of fungi make a huge diverse polyketide compounds.
Right.
Many of them plants do not make them.
Mhm.
So it's that's why the PKS84 mutants still show the toxicity to the planomer.
Exactly.
Yeah, other polycade like PKS. Another question about, I'm just wondering about, you mentioned, you analyzed the promoter. Is the whole like a PKS only controlled by one promoter most of the time?
Well, each gene has its own promoter,
Yeah.
and oftentimes there's a transcription factor in these clusters that will then go and turn on all or a lot of the genes in the cluster.
Mm-hmm.
So hopefully I got the whole, are you asking if I got the whole promoter, or?
There's a particular cluster, you have a cluster over there, right? Not just PKS, because you have so many clusters. So how many, I mean, when you do, you see, you also see many of, when transform factors, many of them upregulated. So my question is, are those cluster gene controlled by one single promoter or multiple promoter?
Each gene in the cluster has its own promoter, but they may have common elements in them that would be targeted by a transcription factor that's also part of the cluster. It
But
depends on the
Your 8-1 data gets to this point,
Right.
which is
There
that only
could be
some of them went on,
multiple
right? Yeah.
transcription factors, right? We don't know that. So a lot of the times there's one transcription factor controlling things, but it's very possible that there are multiple. And so in fact, in this PKS81 cluster, there could be another transcription factor that's controlling the rest of it. So we don't know definitively.
And we see that with aflatoxin. If you overexpress AFLR, some genes are much more highly expressed in others. So it's not, the data are not surprising.
Yeah.
There's probably other thing modulating the individual promoters.
One more.
So in general, though, are a complex like that, are all the genes expressed at the same level? I mean, you need to have that.
Well, not necessarily. I mean, if we remember my slide where I was showing gene expression in the whole cluster in the infected leaf versus in the media. So in those cases, while they were all upregulated, it was to a different extent. But they were all increased in a lot of cases. But it could be that you need different amounts of each one.
You should probably, thank you, Rosalind. It was